if we take neuroscience seriously, then mind uploading is just the logical conclusion of a successful neuroscience. It is no longer a technological or scientific problem about how to preserve brains. We know how to preserve brains. It is a social problem to figure out how to get that available in a quality way to people that really need it.
Thank you for joining the Change I Am Possible, which is India's first future tech podcast. And today I'm delighted and honored to have with me Mr. Ken Hayworth, who's a neuroscientist, president and co-founder of the Brain Preservation Foundation. He is currently a senior scientist at the Hughes Medical Institute's Janelia Farm Research Campus, which is a leading research institution in the field of connectomics. Hayworth is a co-inventor of the Tape-to-SEM process for high-throughput volume imaging of neural circuits at the nanometer scale, and he designed and built several automated machines to implement this process. Hayworth has received a Ph.D. in Neuroscience from the University of California, Southern California for research into how the human visual systems encode spatial relations among objects. So it's a pleasure and honor to have you on the podcast. So let's start with connectomics. Just break it down for the audience what it is.
Yeah, so connectomics is in general the field of analyzing brains at their pretty much their most fundamental circuit level. The idea is that computation in brains and nervous systems in general is done by neurons communicating with other neurons by sending electrical signals between the neurons in the network. And so if you want to understand the computation that a piece of a brain or a brain does, you really need to understand how the neurons connect to each other. So connectomics is a field that whose goal is to map the connectivity so well that they can essentially see how the structure of a circuit gives rise to its functioning.
Right. So people say that a human brain is very similar to a computer. It's an information processing machine of a biological machine of some sort. Would you agree to that?
It's a very dangerous question, right? Because at one level, a human brain is very, very different from a computer. I have an undergraduate degree in computer science and engineering. And so I know how to build computers from kind of the transistor up, and so I can look at the two, how the brain does things and how computers do things, and I can see enormous differences between those. But from a general perspective, the human brain is computational. What I mean by that is that you could. This is a thought experiment. It can't be done today. But if you were to take somebody's retina out and were to replace the retina with a light-sensitive chip that essentially computes the same signals that would have gone down the person's optic nerve. All neuroscientists agree that if you were able to do that, if you were to plug that into the optic nerve so that it generated the same signals going down the optic nerve, the person would be able to see through that artificial retina, and they wouldn't even notice the difference. So you've been able to replace some physical thing with another physical thing as long as it does the same causal influence downstream. I believe that neuroscience in general is committed to the idea that you can make those types of substitutions at any level. So, for instance, there's a glutamate is a very common excitatory neurotransmitter in the brain. But if we were to change all the glutamate to some other molecule, but also change all the receptors that are sensing glutamate to sense that other molecule, there would be no difference. There's nothing magic about glutamate. It's just that causal influences give rise to certain other processes. And so when I say that the brain is computational, I'm really saying that what matters is this cause and effect. Any individual pieces or molecules could be switched out as long as their causes and effects remain the same.
And you mentioned this, that that someday we'll be able to possibly, in theory, we could possibly maybe replace the retina with sending the signals. Is it? Do you think it would be possible in the future to do something like that?
There are major research initiatives trying to do something like that today. The problem is all about the interface between the hardware and the biology, and those are extremely difficult things to overcome in that. Yeah, I think that's probably possible in the future, but the problems involved are not ones about whether the retina is computational. They're more technical problems about how to interface with the optic nerve in precisely the same way as the regular neurons would.
Right. How much do we understand of the human brain at this point of time? And what would it take for us to understand our human brain completely? What would be the timelines?
There's a very simplistic answer that people often give, that neuroscience has just scratched the surface, that the brain is so complex that we have no idea how it works. I think that's completely false. I think we've made, as a field, not me personally, but as a field, we've made enormous progress over the last 100 years in understanding how the brain works. That said, we still obviously can't create artificial general intelligence based upon how the brain works. There are still very fundamental open questions into how representation and decision and all this other stuff works. I would put it this way. What we know is that the level of computation that is important for most of the mental processes that we take for granted in human intelligence, we know that those are resting upon the electrical activation of neurons. So if you look at a neuron, a neuron is an excitable cell because it has sodium-potassium pumps that keep an ion gradient across the membrane, and it has ion channels, and specifically voltage-gated ion channels, so that one signal can be propagated very quickly from one side of a neuron to another side of a neuron, even if that's over many, many millimeters of distance. That's why we can really think as fast as we can. That's why we can interact in an intelligent way in the environment. That electrical sensitivity is then used by creating neuronal circuits that are tied together through very particular synaptic connections. The pattern of synaptic connections allows you to recognize somebody's face. It allows you to make a decision, to move a muscle. It allows you to form and retrieve memories. So it's that level of computation that is occurring, and we can dive down and understand the key molecular and structural components that make up that level. Okay, so I would say that we are 99% certain that most of the computation that we talk about is at that level. Now, from a systems perspective, how does that level combine into the kind of fluid intelligence that we have? That is where we are. We have some reasonably good ideas today, but they are very primitive. that's where we don't understand the brain. We can understand simple things like this is how the brain recognizes a face within a few hundred milliseconds, and we can build deep learning neural networks that kind of do it the same way. But if you want to go the next level up and say, how does somebody take their knowledge in one thing and transfer it to another thing, that's a level of systems complexity that that we haven't yet figured out.
Would you like to share your current updates on your understanding of the Connectomics project or your research that you've been doing?
In the past, I've, as you were mentioning in your introduction, I was part of a team that developed a method of automatically collecting ultra-thin serial sections on tape. That technique has in various forms now spread out to different institutes. So Harvard just published something using that technique that was a map of a cubic millimeter of a little biopsy sample of a human brain. The Allen Institute, with a lot of additional engineering work, converted that tape technique to be compatible with transmission electron microscope, along with Harvard Medical School. And they also were able to show that you could map an entire cubic millimeter. This was part of the IARPA Microns project. So Connectomics has reached the level of mapping cubic millimeter sized volumes with sufficient resolution on the order of 10 nanometers in X, Y, and Z in order to mostly automatically trace out the circuit connectivity. One of the issues, though, is that those techniques have a little bit lower resolution and a little bit less quality of data from an artifact point of view than would really be nice to do reliable automatic tracing. So I've been working on a different technique called focused ion beam electron microscopy. In the team that I work with at Janelia, we mapped a half of a fruit fly brain at 8 by 8 by 8 nanometer voxel size. And again, a whole team of people that are much brighter than me were able to take that data and automatically trace it and get a circuit map of the key central complex circuitry in the fruit fly brain, and then actually interpret it in quite deep ways. So there's a paper that came out that I can barely understand. I just I contributed in some little way in terms of providing this data. That shows how the central complex of a fruit fly brain does vector math in order to do computations of it flying, of it moving relative to external landmarks. So these connectomic projects, I bring that up because these connectomic projects, they really do lay out the circuitry at its, I don't want to say its most fundamental level, because obviously it's made of molecules, et cetera, but at the level that really the algorithms are written. I like to think that, if I had to give up any neuroscience tool, Connectomics would probably be one to give up, if you will, because there's so much exciting stuff that goes on in fluorescence microscopy and in optogenetics and in electrophysiology. Those have really the stuff that we know about the brain has not come out of connectomics yet. But
could you talk a little bit more about that?
Sure. So, I mean, for example, there are papers that I read all the time in the memory engram literature that use genetic techniques to tag a memory, and in the sense of those cells that are active, for example, when a mouse is put in a particular cage and has a fear experience, like having its foot shot, they can tag those cells and then optogenetically reactivate those, or sacrifice the animal and then image the brain to see what those cells are, and to actually see how the connectivity changes between those cells that are forming a memory. They've done some very advanced experiments where they, again, I'm not involved in any of this. I wish I was, but I'm not. Using genetic techniques and optogenetics, they have really started to dissect what that process is, to accelerate the process of transferring things from the hippocampus to the cortex, to show when that can be blocked, like during sleep, etc. There's all these tools. What I'm getting to is there's all these tools in neuroscience that have really started to get to the fundamental questions of how things are represented in the brain, how long-term memory works in the brain, how decisions work in the brain, and connectomics hasn't really significantly impacted that yet, because connectomics can only do small volumes so far. If you want to get all of the circuitry of a region, then you have to do an enormous amount of electron microscopy imaging. So what I am trying to do is I'm trying to develop those tools to get the volume of connectomics large enough and high enough quality so that it can actually address circuits that are large enough to really test these larger theories of how long-term memory and how decision-making works and things like that. So for example, the current imaging technique that I'm working on is something called gas cluster ion beam scanning electron microscopy. And the big difference of it is we have these very fast multi-beam electron microscopes now. Zeiss makes one that has 91 separate electron beams, all focused to about a few nanometer spot size, all scanning a surface simultaneously. And so what we do is we slice brain tissue reasonably thick, about a micron. Okay, it's very reliable to cut it that thick. So we can slice tissue a micron thick, lay it out, collect it onto very flat silicon wafers, and then we put that silicon wafer full of tissue into this multi-beam scanning electron microscope, and it images the top surface of it very fast, okay, at 10 by 10 nanometer pixel size. But then we take that out and we put it in a gas cluster ion milling machine that blasts away the top 10 nanometers, and then we put it back in the multi-beam microscope and image it again. And we just do that again and again and again, and so that 1 micron of tissue is imaged all the way through at 10 by 10 by 10 nanometer voxel size. The reason why that's exciting is because now you get the really high resolution and quality of the kind of focused ion beam technique, but you get it in for high volume, high speed. And you can actually imagine that this could do a whole mouse brain within the next 10 years, I'm hoping.
Wow, wow.
Not that anybody's funded that or anything, but that the idea is to get connectomics to a large enough scale and a high enough quality so that you can really start seeing the exact circuits that are behind these large scale neuronal functions.
Right. So, obviously you said that, at this point in time, you have done a cubic millimeter of a human brain and also mapped a fruit fly. When and if you get to a mouse, what would be the learnings from there, and what are the benefits you think you will see when we are able to understand a mouse brain? And from there, what would be the roadmap to get to a human brain?
So these are two very different questions. One is technological, one is kind of scientific. So I love the question of if you had a whole connectome of a mouse brain. Okay, if you had, and let me be very precise. When I say a whole connectome of a mouse brain, I mean having the entire mouse brain where all of the cells, all the neurons, and the chemical synapses between neurons is mapped. What would you do with that? What would you expect? Why would that necessarily push the field of neuroscience further? Well, it's goes back to something I said before. The level at which we know the least about the brain is not the level of neurons and synapses. We've got a pretty good idea of how neurons and synapses work, and ion channels and receptors, et cetera, et cetera. What we don't have a very good idea about is the systems. And so, for example, you've got multiple modules in the brain, modules in the cortex that are, this part of the cortex learns how to understand visual motion. This one learns how to recognize where it is in a maze. This one learns how to navigate and control decisions. How do those interact? There is an enormous complexity of the systems wiring that allows that to happen. So, for example, learning is the big thing that people are trying to figure out, and how do you do learning in artificial intelligence? Well, the brain is, it doesn't just have a single neural network that does gradient descent learning or something. It has a region of the brain that is set up to learn, and then another region that sends modulatory inputs to tell this when and how fast to learn. It doesn't just have region A connecting to region B. It has region A connecting to region B if region C allows that routing to occur. And so, what I believe is that when we get a connectome of a whole mouse brain, the first whole mammalian brain that anybody would have, we will be able to see the routing between neural modules. We'll be able to see how one module affects how others connect. And that will give us the clues about how general mammalian intelligence works.
Right. And would that help in us building or creating better machines that could be intelligent? Would a understanding of how a mouse brain possibly be a clue to creating?
I think it would. It's really an open question because at one level, neuroscience and artificial intelligence have always kind of worked together. For example, it's like the deep learning neural networks. Are those mainly a product of neuroscience, or are they mainly a product of artificial intelligence research? at some level, it doesn't. They're just connected, right? So I think these like a whole Mouse Connectome project is going to fit into that larger picture as a piece that is indispensable, but is certainly not the only piece that's important.
Right. So you are the founder of Brain Preservation Foundation. Would you like to talk a little bit about that? What is the problem that you're trying to solve over there, and how are you leveraging the learnings of Connectomics to Brain Preservation Foundation?
Yeah, sure. So I am advocating for the development of a brain preservation technique that can be offered in hospitals to terminal patients so that they can preserve their brains indefinitely, in the hope that in the future they could be revived by a very advanced form of neuroscience and scanning, brain scanning, and by ultimately mind uploading. And a lot of my neuroscience colleagues will at that point say, "Oh, well, that's crazy." But the thing is, and I would argue, and I have lots of arguments with my neuroscience colleagues on this. This is really just taking neuroscience seriously. If we take neuroscience seriously, then mind uploading is just the logical conclusion of a successful neuroscience. You know, the neuroscience is one of its core goals is to understand how the brain works at a computational level. Once we understand how that works at a computational level, and once we have the ability to scan a brain and all of the detail that is necessary to get the relevant computational pieces, we will be able to simulate a brain, and that will bring back the original mind that was in that brain. You could argue that yes, this is a possibility in the future, but why discuss it today? I think that's a reasonable attitude to take, except for one fact, that we can preserve brains today so well that it completely covers the range of current theories on how long-term memory and function are encoded structurally in the brain. So what do I mean by that? So the vast majority of high-resolution imaging of large neural systems today, electron microscopy, fluorescence microscopy using antibodies, things like that. They involve imaging tissue that is aldehyde fixed. It's fixed with aldehydes like formaldehyde, glutaraldehyde, things like that. Okay, so you're taking a piece of tissue, you're taking a brain, okay, or a piece of a brain, and if you were to just leave that piece of brain, it would dissolve, it would eat itself, it would decay, right? But if you want to image it, image it at the structural and molecular level, the first thing you do is you put in these chemicals, aldehyde fixatives, that cross-link the proteins together, binding them in place, but not destroying the identity or location of those proteins. And then you can either do fluorescence microscopy using antibodies labels to pick out the individual proteins of interest and image those very precisely at the single synapse level, or you can stain the whole thing with heavy metals and embed it in plastic and do and section it and do electron microscopy. Those are the techniques that we use today to study the circuitry of the brain and the molecular level of the circuitry of the brain. Well, what if we could do that for a whole brain, what if we could preserve it at the same level? So all those molecules and structures that we know are there, because what if we could preserve all of those and then store it for an indefinite period of time? Well, my organization, the Brain Preservation Foundation, we put out a challenge prize to the scientific community and also any other community like the Cryonics community. We challenged them, come up with a method that that could potentially preserve an entire human brain for an indefinite period of time, at least 100 years, so that it preserves those structural features of the brain that modern neuroscience knows are crucial to encoding memory and function. And long story short, somebody won that challenge prize. A laboratory called 21st Century Medicine, headed by a researcher called Robert McIntyre. They came up with an aldehyde stabilized cryopreservation technique, where they took the perfusion with aldehydes that everybody does in neuroscience, and showed that could be made compatible with perfusion of cryoprotectant and then long-term cryostorage at minus 130 degrees Celsius. So the brain is vitrified solid, but is there's no ice crystal formation, there's no macroscopic cracking or anything like that, but it can essentially stay in cold storage at that temperature indefinitely. And then they showed that they could rewarm that. It's dead, of course, because it's aldehyde fixed, but they were able to show that the information is still in it, because we sliced it up and looked at it under an electron microscope and saw that the circuitry, the synaptic circuitry, we could still do connectomics on it. And because this is aldehyde fixed material, basically all the other techniques that that look at the molecules, we know that in principle those would also work as well. Wow. So this is preserving the synaptic connectivity of the brain. This is preserving the detailed ultrastructure of synapses and neurons that we know are correlated with the strength of synaptic connections. But it's also preserving ion channels, it's preserving receptor proteins, it's preserving messenger RNA, it's preserving all of these things that modern neuroscience says are important to the functioning of the brain. And so just to make this very, very concrete, there is a technique that has been demonstrated on laboratory animals in controlled situations, rabbits and pigs so far, this aldehyde stabilized cryopreservation technique that is able to preserve the information content of entire mammalian brains. The organization that I'm president of, the Brain Preservation Foundation, now wants to really start a dialogue with the neuroscience and medical communities to ask the question, how can we take that technique or something better and really get that available to terminal patients that want to use this as a last hope for surviving and waking up in the future.
Right, but wasn't the cryonics facilities, the Alcor and the other cryonic facilities, offering the same kind of preservation techniques, or is this completely different? And if it is, are those companies leveraging it? And does Brain Preservation Foundation has a facility for cryonics, or would you be in the future looking at possibly doing body or brain preservation?
So first of all, the Brain Preservation Foundation is a science advocacy organization only. So we don't preserve anybody. We will never preserve anybody. We don't recommend any existing organizations. The Brain Preservation Foundation is really designed to advocate for the development of these technologies, to really pull it out of the realm of pseudoscience that it has been sitting in, and bring it into the realm of real science. So you bring up cryonics. So obviously this is very, very similar to cryonics. The initial prize that we put forward, I suspected that I had hoped that the cryonics community would win that prize, because after all, they have been preserving brains supposedly for decades. The literature that at least I had seen made me believe that this was a pretty easy task for them to simply demonstrate that the connectome was being preserved by their techniques. They flat out failed.
Wow.
They flat out failed, and they are still trying. I have talked to people within the community that are saying, look, we think that we can use the techniques that we do and show that, first of all, they think that they are preserving stuff, but for technical reasons, they can't demonstrate it. I'm highly skeptical of that, but it doesn't really matter, because the prize was, it was clear that the prize wasn't to convince me, the prize was to convince the larger scientific community with an open peer-reviewed article showing that the connectivity of the brain could be preserved. The aldehyde stabilized cryopreservation technique did that without issue. Every neuroscientist that I've shown those results to, they just simply agree. They're like, oh, well, I mean, first of all, the images look fantastic. Second of all, makes sense that this works. It just makes sense, because this is essentially the stuff that we do all day long, plus cryopreservation. So that technique is easily demonstratable to meet the requirements of preserving the synaptic connectivity of the brain and the molecular details. The cryonics techniques, basically what they're doing is they're saying we refuse to use aldehydes, because aldehydes are deadly chemicals. They're deadly chemicals, and so you couldn't bring the person back without some super advanced technology. That's fine. If they can show that their technique works without the aldehydes, that's great, but they haven't been able to do that. And you can imagine the difficulties. So the difference is that if you use aldehydes, what you're doing is you're chemically stabilizing the brain within just the first minute of perfusing these chemicals. You're locking the proteins in place so that the blood vessels and the neurons, they're all sturdy, and that brain could sit there without decay for weeks, just at room temperature if you wanted to. And that gives you the time to perfuse the cryoprotectant at room temperature to get into every cell uniformly. If you don't use the aldehydes, what you have to do is you have to rush to push that cryoprotectant in while the brain is essentially decaying. And so you have various options, but long story short, you can rush very quickly and get a shrunken, deformed brain, because you're not giving enough time for equalization of the osmolarity on the inside and the outside of the cells, or you can take a longer period of time and you got a bunch of dead, decaying cells. And so the cryonics as it is practiced today, my understanding, I don't know how well it works, but I know that they have not demonstrated that that they can preserve even the bare minimum connectivity of the brain.
Right. So you saying the technique that the company which won the prize 21st Century Medicine can not only does not only show that it can completely preserve all the information of the brain, but eventually, maybe in the near future, 50 years, 100 years, you will be also be able to revive the cryonically preserved the brain, right?
No, I don't think so.
Yeah, please
So, well,
explain.
let's unpack this. So the technique that this research company demonstrated on rabbit and pig brains, this aldehyde stabilized cryopreservation technique, I have no problem saying that it preserves the information content of the brain. But I have to put a qualifier on that. And here's the qualifier. It preserves those ultrastructural and molecular features of the brain that modern neuroscience says encodes the information in the brain. Could be that there's something that the neuroscience textbooks don't know about, and that, you know, since we don't understand the exactly how the brain works, that's always has to be an option. But I will say that it preserves, you know, the synaptic connectivity, the ultrastructure of synapses and neurons, and the key molecules like ion channels and receptor proteins that are thought to be important in neuronal function.
Right.
But it preserves it in a way that uses aldehyde fixation, and so it is totally not able to revive that, because you're gluing all of that stuff. It's preserving the information while destroying the biology, the biological function. In fact, those two things are being tied together. You know, it's essentially it's such a good technique because within the first minute, it glues together all the stuff that could be decaying. And gluing together proteins means that it's, you know, it's I don't want to say it's an impossible task, but it's an almost impossible task to unglue things and get them working again. But if the information content is preserved, which I'm arguing, that should make it compatible with very advanced brain scanning techniques that could allow for future mind uploading.
The neuroscience community, I think a few years back, demonstrated how a C-Elegan worm could be entirely captured and be uploaded onto a computer, the Connectomics project. So in theory, if you
I disagree with that.
Yeah, please do yeah, talk about that, because
so we do not understand as a neuroscience community how C-Elegans works enough to claim to have uploaded as C-Elegans. I'm just, it's a correction. There's some modeling that people have done that might be the bare minimum of showing that you can take some connectivity diagrams and show some behavior, but there is, you know, I know people that are working in the C-Elegans research community. It is far from being completely understood at a level to allow computational simulation of the C-Elegans brain.
So you saying we like really far away, but you know, at the other side, you know, there are these entrepreneurs who are working on trying to build these brain computer interfaces. There's obviously, everybody knows, you know, the Elon Musk. There's companies like Facebook who's invested a large amount of money in R&D, and you know, they have an offshoot called Control Labs where they're actually building these wristbands which can eventually be an interface between augmented reality, virtual reality. And then there's Kernel, which Brian Johnson is heading. So what do you say to these companies? Where do they stand, and when do you think that like we will have like a working brain computer interface which will be able to create a interface between a human and a computer?
Yeah, I have to say that don't have any expertise in that area beyond just, having a Ph.D. in neuroscience gives you some some level of understanding, but the intricacies of brain-computer interface are beyond my expertise. I will say though that we've had brain-computer interfaces for a long time in the neuroscience community. It's just that the interface between the brain and the hardware is very difficult, and so I'm excited about the Neuralink type of technique because it could be very very robust. It seems to me, again, I'm not an expert in this. It could be a longer-lived technique where you can really implant this and have it last for years and years and years, and you should be able to. I think the most exciting thing, you're going to hate this, but the most exciting thing from my perspective is all of these neuroscience experiments that we currently can only do on animals could be then done on us, like me. I mean, if it really becomes like everybody's getting it or something, I don't want to be the first volunteer or something, but it would be wonderful to be able to kind of read off a pattern and have another pattern be imposed on your brain and feel what that is like. What is it like to have a thought put in by a brain-computer interface? And I'm not saying even I'm saying from the perspective of a neuroscientist that has really wanted to understand how the visual system, for instance, processes things. Having that ability would be enormously helpful to figuring out how the brain works. So my take on brain-computer interfaces is it would open up an enormous space of experimentation in humans, ethically, that we simply don't have today.
Right. Yeah. Obviously, I guess once we have like a working brain-computer interface, there'll be so many ethical, moral, and ethical implications that might be open up. You are an advocate of mind uploading. Is there some synergy going on within brain-computer interface? Because obviously, when you have like a completely working brain-computer interface, that means that you can actually understand the working of your human brain, the structural and the functional capabilities of your brain, and that's the only time you will be able to map it to your computer, to bits. Where does the mind uploading stand? Where does this? When do you think it becoming a reality, and what will it take for it to become a reality? Is there imaging technique? What would be the process?
First of all, I don't think brain-computer interfaces have. I don't think that's a route to mind uploading. There are some people that think that might be a route to mind uploading. I do not think it's a route to mind uploading, and I'll tell you why. So if you look at how the information is stored in the brain, what makes us unique individuals, it's not stored at the individual neuron level. It's stored at the synaptic connectivity. And a brain-computer interface is just listening to, at best, individual neurons firing. So it is not easy to infer all of the information that is stored in a network by just listening to hours or days or whatever of a recording and trying to figure out how things are, what's actually in there. If you're able to actually scan the brain at this synaptic level, then I would argue you do get everything. All of the memories, all of the different types of memory, perceptual memory, declarative memories, procedural memories, all the innate circuitry that you're born with. All of that stuff is there at the structural level. And I think that is what is necessary for mind upload. So let me give you a little bit of a, so if I had to wildly speculate, the first thing to understand is, again, we're nowhere close. We're nowhere close. It is a long, long, long haul of neuroscience. And you know what neuroscientists should be doing right now to best get to mind uploading? They should be doing exactly what they're doing. Okay, they're doing basic neuroscience research, trying to figure out how the brain works. Okay, this is exactly what the neuroscience community should be doing on that long road toward mind uploading that may be centuries off. Having said that, though, if I had to speculate on how we're going to get there, I think that there's all of this exciting research that is the genetics, optogenetics, and calcium imaging. Lots of stuff that is in the living animal and in the living human understanding of how the brain works. Lots of fantastic computational and theoretical work. But then you got this connectomics piece that is going to get larger and larger volumes and better at mapping circuitry. And I see that we're going to have a fruit fly brain. We're going to have cubic millimeters of brain. We already have these things, essentially. We're going to get better at that. We're going to get better at understanding those circuitries, but we're going to reasonably soon be able to get whole hippocampus of a mouse or something. We're going to, within 10, 15 years, I would speculate, be able to do to get a connectome of a whole mouse brain. This will continue until at some point this century, people will decide that they want to do a connectome of a whole human. I don't know what technology they will use, but there's at least the electron microscopy ion milling approach. It could theoretically be scaled all the way to a human brain. There's nothing that's stopping that.
Right. So who amongst your peers you feel are asking the right questions, and would you I mean, do you have any views at what Henry Markram is doing with the Blue Brain project? His in silico way, where he's trying to build it, like in the computer, simulate a brain in a computer.
I mean, I don't want to say too much on that, but I agree with most of my neuroscience colleagues that I've at least talked to that. The simulation that he was doing was way premature. Essentially, that project was designed to simulate things where there was a lot of missing information about the connectivity. He made some arguments against not needing the connectome. I didn't buy those arguments, and I haven't seen anything that that has shown that really good, solid new knowledge has come out of this. I'm sure there's some. It is the approach that I would take. In terms of other neuroscience research, I read the neuroscience literature voraciously. I read all the time. I love it. If that's all that I could do, I would just keep reading neuroscience papers. Okay. The neuroscience community is fantastic. It is. There are so many interesting threads that people are following. I think they are asking the right questions. I don't even know where to start in terms of them, but I would point the ones that I have that I read most with most interest nowadays are the ones that are trying to pick apart these memory systems. But there are other things like there's the hippocampus system. where you're trying to figure out how the hippocampus represents and learns to navigate arbitrary spaces. There's this fantastically detailed experimental work that is able to, using electrodes or using optical imaging, is able to decode the patterns of activity and show that, oh, these patterns of activity mean that the mouse is here and it's about to do a trajectory like this. But you can also look at that same area, that same hippocampus, while the mouse is sleeping, and see that it's replaying at hyper speed the things that it has done during the daytime. So all of that experimental research is being matched on the computational side by people that are putting together really advanced models of how neural networks that are biologically realistic are doing these types of computations in the hippocampus and other brain regions. So this is making significant progress toward the key question of how the brain works. At the same time, you can look back at it and you can say, well, that's just the hippocampus, and you know, the entire system uses this information in a super flexible way where it can do one-shot learning and decisions that those little models certainly can't do, and none of our artificial intelligence can do. Again, it's a long haul. We understand this little module a little bit better. We understand this little module, but we don't understand how they're connected together. The experimental research work is progressing, but it has, it has limits in how many neurons it can record from, and what temporal resolution it can do. The Connectomics is progressing, but it can only do cubic millimeters for right now. All of this stuff in the next few decades, they're going to come together. Right. And I believe what you're going to see is you're going to see the Connectomics produce large enough volumes of circuits that you can look at those circuits and see which of the computational models are right and which are wrong, and move the theoretical stuff in the right direction.
Right, yeah. So we're getting into a very exciting space. I'm going to digress a bit. There's these bunch of philosophers who've been saying that free will does not exist. In fact, there was an experiment done which is known as the Libet Experiment. Would you like to talk a little bit about that? Do we have free will, or is it just an illusion?
I think it's semantic. That's my humble opinion on it. When I think of myself, I am maybe this is obvious. I am completely comfortable with the idea that I am a brain. I am a computational mass of neurons, and when I ask questions about free will and identity, I can put them in that perspective. So, for instance, the free will problem, I'm not even sure if I have an opinion on it, because to me it's just obvious. It's like, well, does C-3PO, the robot in Star Wars, right? Does C-3PO have free will? It's like, okay, well, he's got a computer program and a robotic brain, and it does what it does, but he's the one that's making the decisions, even though it's his program, because he's his program. There's no separation between the two. So if there's no question about C-3PO's free will, then how could there be a question about my free will? Because I'm just a robot with a computerized brain, but it's made of different things, like biological neurons. It's the same answer. is what I'm saying.
I'm vested deeply in virtual reality. I've been building virtual reality content, and virtual reality is the tool that even now it can trick your motor cortex into believing that the virtual is real. There's these simple game where you put on the VR headset and you get into a building. You get on the top of the terrace, and on the top of the terrace there's this plank which is out. And in a virtual reality world, and everything is animated and it's cheap animation. But as soon as you step on it and you walk, it tricks your motor cortex into believing that standing on the top of the building and not in your house. So I think we are in this next few decades. There is going to be great transformation. There is artificial intelligence, which is the field is accelerating. We're getting towards AGI. I'm not saying that we reaching AGI. It could be possibly, I don't know, maybe like 50 years or less. But all this seems to be possible in a human's time horizon. Then there is the other field where the genetic editing and things like that, where we're working on human longevity. And stuff like this. So we are in an exciting space. So I personally am super excited about what technology can do to us in this decade and more. You have been vested in neuroscience, and you spent your life on the trying to understand the brain. What is it that excites you most, and what question, if you get the answer of, you think will be like great for neuroscience?
I think the thing that really excites me the most is that that future, we have always considered it as either we're going to get a little slice of time and then we're going to grow old and decay and die, and we're going to miss out on everything beyond that, right? Or maybe we'll be lucky enough to be the in the group that finally cures aging. Okay. So first of all, I'm very skeptical of the ability to cure aging within my lifetime. Certainly. Even within my kids' lifetimes, I think that it is highly unlikely. It's not an impossible. It's just these things take a long time. The message of brain preservation is that we can do brain preservation. That is a technically solved problem. It is not a socially solved problem. It is not available. Nobody is making it available. Nobody is doing it the right way. What we're trying to do is we're trying to get the scientific community and the medical community to actually develop this like any other surgical procedure and make it available in hospitals in a regulated manner. Make it available, and there are many more ways to screw up a brain preservation than to get it right. Okay, it is a technically difficult thing to do. It can be done, but it's a technically difficult thing to do. And so what I would maybe request your reviewers to think about is that enthusiasm for the future. How much more would it be if there was a reliable, guaranteed brain preservation option for everybody at the end of life, so that there was a very good possibility that if you wanted to, that you would actually wake up 200, 300 years from now when all of this technology is developed. I think that is the world that we can get to if we fight for it, because it is no longer a technological problem or a scientific problem about how to preserve brains. We know how to preserve brains. It is a social problem to figure out how to get that available in a quality way to people that really need it, to people that are enthusiastic about the future and don't want to just give up on the possibility of enjoying it, the real far future personally,
right,
in a healthy way.
Thanks a lot for taking time talking to us. And I guess we deserve to choose, and if we have to choose. There shouldn't be a social group opposing it. I mean, we should have the right to choose. And thank you for not over glamorizing future tech or everything, and giving us the way it is, fact rather than fiction. So thank you for being part of the podcast. And to my listeners, if you like what you see in here, please press the subscribe button. Until next time, see you guys. Bye bye. Thank you.
Thank you.